Bioengineering discussion. Today you will see 7 video clips that explore modern artificial selection through bioengineering. After each clip, be prepared to discuss and write your response to share in class.
In this day and age, it's not uncommon for we humans to try and invent new things that benefit us. But genetically modifying animals, really? That's right, even Mother Nature isn't safe as we alter everything we can for the greater good. For this altered installment, we're building a zoo of 10 unique genetically modified animals. We promise you, number 1 should be a shocker. Number 10, glow-in-the-dark cats. Inserting rhesus macaque and jellyfish genes into unfertilized cat eggs produces a fluorescent kitten resistant to feline immunodeficiency virus. More than just a convenient nightlight, the Mayo Clinic scientists responsible for this line hopes to use it to fight the human strain of AIDS. Oh, by the way, speaking of pigs, number 9, Popeye pig. This specific type of pig is created through the introduction of a spinach gene into unfertilized pig eggs, which are then implanted into a surrogate. These pigs are born with no health complications and produce a protein that is normally found in spinach leaves. Number 8, Mexican salamander. The restorative properties of the Mexican salamander has been under the microscope for some time now, with scientists genetically modifying these tiny creatures to get a better understanding of the regenerative growth of a lost limb. Thanks to the modified salamander, scientists have deduced that cellular growth doesn't start at the beginning, but rather at a more mature version of the cell, giving hope that the same process may be possible in human cells. Number 7, curative camels. That's right. Dubai scientists have been hard at work genetically modifying camels to produce milk rich in pharmaceutical proteins to be later used in the production of insulin and anti-clotting agents. Number 6, Andy. Andy, an adorable rhesus monkey, was born in October of 2000 with the sole purpose of creating primate models of common human diseases such as Alzheimer's for research and drug testing. Sounds like quite the life, right? The process to create Andy started with 224 unfertilized eggs, all injected with a virus carrying the green fluorescent protein gene, an easily monitored gene due to its green glow. Of the 224 eggs, Andy was the only of 3 surviving specimens to carry the gene. Number 5, Mighty Mouse. They're like your average house mice, except that they can run up to 3.7 miles or 6 kilometers per hour continuous 6 hours straight. The mouse is capable of eating 60% more than an ordinary mouse, but is more likely to remain leaner and more muscular. So, why do we want a breed of super mice? According to the researchers involved, these mice could help pave the way for the development of muscle enhancers to assist with certain conditions. Or, Number 4, sudden death mosquitoes. Oxitec, a United Kingdom biotechnology firm, engineered a breed of mosquito that is either sterile or will die before even reaching adulthood. The aim was to cut down on mosquito-borne illnesses, including dengue fever, but there was an unexpected side effect: the complete eradication of the mosquito population. As with most things in nature, even mosquitoes serve a purpose, such as being a meal for a variety of birds and fish. While we may love the idea of a land without mosquitoes, there could be detrimental effects on the local ecosystem. Number 3, featherless chickens. Scientists at the Hebrew University in Rehovot's agriculture department engineered the naked chickens as a means of creating a low-fat poultry that also happens to lower the cost on ventilation systems for farmers. What the featherless chicken really determines is that feathers are actually quite important to birds, protecting them from temperature fluctuations, parasites, sunburn, and mosquitoes. Number 2, web-spinning goats is the product of incorporating the spider dragline silk gene into goats so that our four-legged friends would produce the silk protein in their milk. So maybe we jumped the gun with spider goat, but this genetically altered goat saves time and energy, as researchers won't have to deal with the spider farms in order to produce the silk protein, which is used in artificial ligaments and tendons and jaw repair. Number 1, babies. Perhaps it's not as bad as it initially seems. Genetically modified babies are not really new to the game, and it's actually a lot less severe than it sounds. Sadly, because who doesn't want a team of super babies running around? The concept of modified babies actually refers to the process of cytoplasmic transfer, which entails injecting cytoplasm for a donor egg into one lacking mitochondria. The egg then goes through a process of fertilization and implanting into the womb, hopefully creating a baby that technically has 3 parents.
Discuss the top 10 genetically modified animals, then write down the one that your team is most concerned about. Be prepared to explain.
This googly-eyed 5-month-old pug is named Momo-tan, and for now she's living amongst dozens of other newborn puppies. But these aren't your standard run-of-the-mill puppies. They don't know it, but Momo-tan and each of her puppy companions are clones. For 16 years, Mr. Fukuda was the proud owner of another pug named Momoko. The two were inseparable. When Momoko recently died, Mr. Fukuda wanted to bring her back, so he turned to a company called Suam to recreate her.
Currently, we've cloned more than 600 dogs of various different breeds, and there are no particular limitations to the different breeds.
Suam claims it can clone any dog, no matter the age, size, or breed.
Would you want a clone of your dead pet? Discuss your answer with your team and be prepared to explain.
Closer to human clones. The question has been raised after scientists in China produced 2 genetically identical monkeys. Zhong and Hua are the first primates to be cloned using DNA transfer, the same technique which produced Dolly the sheep 20 years ago. And now scientists plan to create more genetically identical monkeys. And we're talking populations, not just 1 or 2. They say these monkeys will help with future research into human diseases, but campaigners fear the experiments could lead to human cloning, an idea that raises many ethical and moral questions. Activists also claim the tests involve unacceptable levels of animal cruelty. The scientists have even allegedly admitted that their work has resulted in the abnormal development and death of many monkeys. Zhong and Hua were the result of 79 attempts. The cloning process involves transferring cell nucleus DNA to a donated egg that is then prompted to develop into an embryo. Sheep, cattle, pigs, cats, mice and rats have all been cloned with this method, but it's the first time for a monkey. So the question on many people's lips, how long before we see the first human clone?
Would a clone of a person be a real person with all the rights of anyone else? Discuss with your team and be prepared to explain.
Adaptation and selection. These 2 principles have molded basically all life on Earth, from pond scum to platypuses. Evolution's rulebook says changes to life's instructions happen more or less at random, and not because an organism needs something. While selecting which traits are good enough to be passed on is decided by, the universe. But humans are rewriting these rules in incredible ways. putting us in charge of our evolution. For starters, we've massively changed our environment, so it's much harder to die. There's almost no chance a predator or another human will kill you today. Science can repair our bodies or make us invincible to enemies we can't even see. And more people die today from having too much to eat than too little. For thousands of years, we've been deciding which plant and animal traits are useful, which are passed on, and which are not. Thanks to more and more selection under our control, human life expectancy has doubled in just a few generations. But this simply lets us steer evolution. Once we discovered the molecule that guides these traits, we realized we could hit evolution's gas pedal. DNA's information is encoded in long chains of nucleotides, and natural mutations, good or bad, happen very rarely and in random spots. In the mid-20th century, scientists realized they could speed up mutations using radiation or chemicals, but the effect was still up to chance. And contrary to popular belief, never, ever leads to superpowers. Later, in the 1970s, scientists exchanged entire chunks of DNA between species, creating microbes that manufacture insulin, plants resistant to viruses, even mice with human genes. They could control what was being edited, just not where in the genome this new DNA was inserted. What scientists needed was a tool so precise it could make a single change in 3 billion DNA bases, and cheap enough that anyone could use it. In 2012, they got it. CRISPR. It sounds like a breakfast cereal, but CRISPR has the power to literally reshape humanity. And by reprogramming the target, CRISPR could cut any genetic sequence down to a single DNA needle in a 3 billion letter haystack. Suddenly, we have the power to edit genomes like a word processor. When cells repair cut DNA, they can glue the ends back together, often trimming a letter or two out and disrupting the genetic code. Or the cell can use another template to write in new DNA, letting us splice in new genes with surgical precision. Genes can be switched on or off. Infections like HIV cut out. Our own immune system can even be reprogrammed to hunt down cancer cells. Thousands of human diseases are caused by mutations in single genes, and each could be reversed with CRISPR. Even complex traits like height or heart disease caused by many genes interacting with the environment in ways we don't fully understand might be within reach.
What do you think is the most exciting thing that technology like CRISPR can do? Discuss with your team and be prepared to explain.
That's exactly what you think it is. It's poop. At this farm, manure is money. One of Florida's largest dairies is turning it into energy.
Manure is not a waste here. The cow is the ultimate recycler. She makes milk, and as she's making milk, she's making manure. That manure is then used as fertilizer to grow crops that we harvest and then feed back to her. One area where we weren't capturing the value is the methane production that can occur if you put manure into a digester. 70% of our electricity that we use on farm we now generate ourselves, and then we'll sell the excess to the grid. Our generator is 1,000 kW, and so that is equivalent to powering 425 homes.
Do you think cow poop will be the energy of the future? Discuss with your team and be prepared to explain.
Mice are creepy enough, and this one has no hair and a human ear growing on its back.
Yes.
He doesn't seem to mind that he has an ear growing on his back.
No, he knows he's here for a bigger purpose. But this is a very, very important step in the science, because on the back of this animal, we're actually incubating and growing perfect cartilage in the shape of a human ear. And it's completely connected to the blood vessels, so that it's just like a native ear in a normal circumstance.
In the head of a person.
That's correct.
So when this finally gets implanted in a human, you don't expect rejection, as is so common with new body parts.
Exactly, because we're going to start with the patient's own cells. It'll make his own tissue, and therefore the body will accept it.
Within a year, Vacanti and Langer expect to be implanting their ears directly on the heads of soldiers wounded in Iraq and Afghanistan.
One day, any organ will be able to be replaced by genetically engineering them in a lab. Is this a good idea? Discuss with your team and be prepared to explain.
In the last several years, we've seen some strange genetically modified organisms. Pigs that glow in the dark, chickens that grow without feathers, and even goats that produce spider silk proteins. But what really gets people riled up are genetically modified foods. The prospect of eating somebody's science project turns the stomach of those who aspire to eat so-called whole or natural foods. But what exactly does genetically modified mean anyways? And how are those foods created? To answer these questions, we need to go back in time about 10,000 years. That's when people first started domesticating animals and cultivating plants for food. By picking organisms with desirable traits and mating them together over and over again, people were able to mold species to meet their dietary needs. The classic example is corn, which started out as a tropical grass called teosinte, with small, nearly inedible kernels. Over thousands of years, ancient farmers in Mexico transformed the grass into the delicious, starchy treat we like to eat today. Scientists call this process artificial selection, and it's made all kinds of unappetizing plants edible, from wheat to rice to almonds and bananas. What's different about modern genetically modified foods is the speed and precision with which we can make changes. Now, instead of selectively breeding for traits, we can go directly to the DNA responsible, snip it out, and transplant it into newly developing plants and animals. If these snippets come from the same species, scientists call the new organism cisgenic. Cis means the same. But if the DNA comes from a completely different plant or animal, scientists call the new organism transgenic. Trans means to cross. Those fluorescent pigs, they glow green because they contain a gene from a luminescent jellyfish. And in the last 20 years, we've begun to see the food equivalent. Corn that makes bacterial toxins poisonous to pests, tomatoes that make the antifreeze proteins from fish, and even cows that produce human milk. So you may be wondering if this kind of overt manipulation is safe. Well, all of the GM foods currently on the market have been thoroughly tested, but the verdict's still out on some of the more exotic transgenic pairings.
Are you concerned about eating GMOs, genetically modified food? For example, would you eat a tomato that glows? Discuss with your team and be prepared to explain.